Hello, my name is Mr. Johnny Hopkins, and welcome back to What is a Biology? Today, we're going to be talking about something called transcription. And if you remember anything about the first video, something called the central dogma, this is the first part of it technically. Transcription is going from DNA to RNA. Just that simple. And so, how do we go from DNA to RNA? Well, you need to take that little guy at the very end of the central dogma, a protein. And this protein is called RNA polymerase. There are multiple different kinds of RNA polymerases, but we're just going to, for all intents and purposes, pretend that it's just going to talk about the family and not contain specifics. You can do that on your own if you really want. So, what RNA polymerase does, believe it or not, it polymerizes RNA. So what it does is it binds to the DNA, it locally melts the DNA, and polymerizes RNTP into an RNA chain. So, first of all, what do I mean by melt? See, melting is actually a very ambiguous term here. What it doesn't mean like it's boiling something down into making it go from the witch to a puddle. No, what it just means is it essentially untangles the two DNA strands. So, like in this figure, you just see one DNA strand kind of go away from the other one. It just means that they kind of get uncoupled for a little bit in a little tiny thing called a bubble. Then it polymerizes RNTP. That just means it kind of stacks one on top of another. It just makes it into a polymer, into a wiggly boy. So, what's RNTP? RNTP is something called ribonucleoside triphosphate. So, as we remember, we have a nucleoside, which is a base and a sugar, pentyl sugar. So this is the nucleoside. Triphosphate, tri means three, like the Triforce. And you have phosphate, phosphate, like these guys. You have a single phosphate here, and so on and so forth. So, 3 phosphates, triphosphate. And one thing that we're going to say back again and again and again is that these NTPs have a lot of energy storing capabilities, such as we'll see in translation that they have ATP and GTP involved, which makes some very fancy stuff happen. Makes me all excited. So you got nucleoside, triphosphate. Awesome. With those 2 OHs, which is particularly important for RNA. So, we're just going to look at this little figure real quick. You got RNA and you got DNA. DNA is disrupted by this RNA just binding in, and it makes a nascent RNA chain. Nascent just means growing. It's growing, it's growing, it's growing. It's like the child you raise. They just keep growing and growing and growing as you get older and older. So, this nascent transcript comes from a DNA, and what is made is it goes from it's made from DNA from 5' end to the 3' end. So what that means is because you have to couple this RNA polymerase starts the transcript at 3' end and makes its way down to the 5' end of the RNA. So it locally binds to 8 base pairs, but the bubble, this bubble that's the locally melted DNA, is 12 to 14 base pairs long, depending on what point in the sequence and what polymer you have. What polymerase you have. So how does this guy just do all this stuff and make all this RNA so we can get some proteins in here and get some real action going? And the way that happens is you got 3 steps. Initiation, elongation, and termination. Initiation, very, very simple. RNA, go to DNA and says, hey, I'm going to hug you. We're best friends, and I'm going to run along you and try not to tire myself out. So RNAP will bind to a region in the DNA called a promoter. Promoter is just a region in the DNA that usually is at the beginning of the gene and has something called transcription factors attached. Transcription factor is a protein. It's just a protein that binds to DNA and binds to the promoter. So factors are just factors are proteins that just bind to DNA. And so what their job really is typically to do is to either just find RNA polymerase and pull it towards them. It just kind of grabs on and says, hey, you should do this. You should do this right here. You should really do this. And then that's able to make some RNA. And it grabs RNA, takes it to the promoter, and then RNA polymerase will go, grab two RNTPs, and then, hey, elongation begins. So now we're at step 2. Elongation, elongation, elongation. Just keep going, going, going like an efficient train. And so one thing to remember is that RNA binds to DNA, and it does so perfectly in the Watson-Crick base pairing structure, such that you have adenine bound to uracil, cytosine bound to guanine. So A to U, and T to A, C to G, G to C. So then you have it elongate, forming these Watson-Crick base pairs and adding on these RNTPs. How does it add on these RNTPs though? The way that it does is by using this. If you remember, when we create an RNA or a DNA chain, they have only one phosphate. And what they're able to do actually here is they shoot off this second one right here, this guy that's labeled beta, because what happens is, so you shoot off this beta phosphate, and that gives you a bunch of energy. It's all energized, it's ready to go and do some major damage, and just kind of just joins on and holds on with its life to this new RNA chain. And so that allows it to drive this force. A simple way to think of it is you have all this electrostatic energy by all these negatives right next to each other, and all that negative energy gets shot out, and so because it goes into positive energy and recites some mantras, that allows it to form its yoga circle of its RNA nascent peptide chain. So it just kind of keeps going on from the 5' to 3' end of the DNA, making RNA go along the 5 and finish at the 5'. So now we can come into this concept of a ribose switch. So as RNA is formed, RNA can base pair with itself and form secondary structures. And sometimes different RNA requires the formation of a ribose switch, and it's just this regulatory region of a nascent peptide, of a nascent RNA chain. And effectively, what can happen is a RNA polymerase will, the RNA comes out as a wiggly boy, and if it forms into a compact secondary structure, it can bind something else. Typically, it can be like an RNTP, and then it will bind that, and then elongation will keep continuing. But sometimes the wiggly boy will stay a wiggly boy and not make a proper secondary structure, not bind something, and then just the whole process terminates. Which brings us to the third part, termination. RNA either can preemptively, such as in the case of the ribose switch, or finish transcription and just dissociate, so we have a final so we have a final RNA transcript. And that guy can do some real major awesome stuff by going out into the world and making some proteins. Gets all excited. Can become Sonic the Hedgehog one day. So now we come into this concept of supercoiling. So we're going to talk just more in general about things surrounding RNA and polymerization. So supercoiling, the simplest way to just think about it is in the bacteria. So in the bacteria, the DNA is just kind of like one giant circle. And it's just circle inside the cell. And what happens is when you have these guys, it pushes and makes this weird bubble, and that actually causes it to twist in on itself. So like, just think of a rubber band, if you twist it a bunch, it comes into this weird guy. And then you need to take it back and unwind it. But when you have this bubble, it's not very easy to do it. So you got these things called topoisomerase. And this does not have, supercoiling does happen in eukaryotes as well, which have the chromosomes, which are not circles, but that's just because they're all kind of linked to the membrane and such with different filaments, such that you have anchoring that causes it to fold in on itself, like shown here. So topoisomerases help this guy happen, help this guy relax. So you have topoisomerase 1, which cuts it. It cleaves the supercoiled DNA and relieves the stress of supercoiled DNA. And so then it just kind of unwinds and unwinds and unwinds and goes back to this, but it's got that cleave still. So you have topoisomerase 2 come and join in on the party. And what topoisomerase 2 does is it ligates the ends of the cut by topoisomerase 1. So what these guys allow to happen is for a supercoiled structure to go back into being a normal, stressless environment. So, that's RNA and DNA in a little nutshell. Now, I'm going to go into a little bit of that. So, an operon is an arrangement of genes into a functional group. That really means nothing, but it's just a word you're going to hear a lot. It's another Socrates thing. So, RNA has this little fancy thing called introns and exons that you've probably heard of. Intron is a non-coding region of the RNA. Exon is a coding region of the RNA. And in general, introns are called untranslated regions, such that like when you have the RNA turn into a protein, you can write down a bunch of words on paper and just cut out different segments and try and make into the best possible story. So you want to edit out words that shouldn't have ever existed there. You can say nonsensical sentences such as a dog ran after the cat. So you can get rid of watched after the cat, watched ran after the cat, get rid of watched, a dog ran after the cat. Makes sense. So that's kind of the idea of introns. You edit and delete out certain segments that shouldn't exist in the RNA so you can have proper formation into a protein. So, another interesting way is that untranslated regions also can be added onto the DNA, onto the RNA right before. So, like, think of it as kind of like a table of contents and an appendix. So you have RNA, which is extremely volatile. It can degrade very instantly in just this native state created from the RNA polymerase. It's kind of as much useless. It's gone. It's dead because of those OHs right here. They allow for nucleophilic attack. It's bad. It's bad. You're gonna get nowhere if you don't protect it. So what eukaryotes have done is you protect it. They teach it proper sexual education, and they teach them protection. So what happens is, you have the 3', you get this poly A chain. So it's just, what happens if you just add a bunch of adenines to the very end of it, such that if you, some fancy, smug-looking protein comes by and is like, hey, I see this RNA, I want to beat it. So it takes out the final adenine and thinks, I did a good job here. That guy should have been degraded. But then, it just took out one of over 1,000 that can be added. And then you have the 5', we have a 5' cap, which is around 100 nucleotides, which also protects the main segment in the middle that was created by the RNA polymerase. So now we have this concept of splicing. So as we talk about introns, there are coding and non-coding regions of the DNA, and we need to get rid of those regions that shouldn't exist there. And what happens is you have something called RNA splicing. And splicing is the removal of these untranslated regions, which can be done by proteins or by the cell. And what that means is that RNA just kind of folds in and it catalyzes its own reaction and gets rid of these introns. So you have alternative splicing as well, which is an even fancier technique, which is done by actually 60% of genes, so it's very, very common. And what you can have is like 3 exons separated by 2 introns, and you can actually have this thing alternatively spliced such that the first 2 are ligated together, but the third is missing. The first can be ligated with the third, such that the second is missing, or the first and the second and third can all be present. And this gives a different protein, which gives different diversity, which allows the cell to respond properly to everything. One notable example is fibronectin. You have 2 different variants that can be caused by alternative splicing, E3A and E3B, and when they are not both present in the cell, you do not get clotting in the blood when you start to bleed, which is very, very bad. So fibronectin, by having both alternative splices, you get proper function of the body, and so you don't literally bleed to death from just a paper cut. So that is everything on cover. We're going to just real quick summary. RNA polymerase goes from the 5' to the 3' end of DNA, making RNA from the 3' to the 5' end. And so it initializes it, it elongates, and you can have ribosomal switches form, which help modulate whether or not the RNA chain actually finishes. And then it terminates once the mRNA is complete. You have supercoiling, which is where the RNA folds in on itself like a rubber band, and topoisomerases relieve that stress. Then you have introns and exons, which can be coupled with the addition of a 5' cap and a 3' poly A tail to these proteins, to these transcripts of mRNA. And then you have RNA splicing with alternative splicing, where you remove the introns inside the segment such that you have only the exon, so you can have proper different diversity of proteins exist by stringing together different domains. So that's all I wanted to cover today. I'm Mr. Johnny Hopkins. This has been What is a Biology? Thank you.